All right, well, welcome back. We're ready to start our penultimate session here, multi-omics integration. The next session after this will be predictive approaches that we'll transition straight into after the Q&A following this session. Both these sessions are just 2 speakers each, and then we'll have concluding remarks for the meeting. Again, thank you all for being here. We appreciate it. Thanks to our speakers for sharing their time with us and their thoughts. We're excited to hear about these last 2 sessions, multi-omics integration and predictive approaches. So I'll go ahead and let Vanessa take it away for multi-omics integration. Take it away, Vanessa. Thanks.
Great. Thanks, Keith. I'm excited to be here today to share the TSMC Bioinformatics Multi-Omics session. My name is Vanessa Varaljay. I am the Bioinformatics lead for the Biological Materials and Processing team in the Materials and Manufacturing Directorate at AFRL. So we know that combining multiple omics techniques has become increasingly popular, and it a broader, more holistic perspective of microbial systems than traditional techniques. This interdisciplinary research approach using multi-omics data and modeling can be challenging, but I think with the right tools and methods will revolutionizes how we will ultimately uncover microbial communities and their interactions and influences on ecosystem outcomes, especially what you hear today in terms of disease and effects on the host. So in this session, we have 2 fantastic speakers, Dr. Keith Crandall and Dr. Sean Gibbons. We're going to share their research and innovative approaches for analyzing multiple omics data through a systems biology lens. So with that, I would like to introduce Keith Crandall, who is a professor and director of the Computational Biology Institute at George Washington University. His lab focuses on the development. Sorry.
Sean's going first.
Oh, I'm sorry. Let me switch gears here. So Sean Gibbons is an assistant professor and the Washington Research Foundation Distinguished Investigator at the Institute for Systems Biology in Seattle. His lab studies ecology and evolution of host microbial systems and how commensal microbial ecology influences host phenotypic variation. So with that, Sean, thank you for being here today, and I'll let you take it away. Sorry for the mix-up there.
Oh, no worries. Thanks so much, Vanessa. Let's see if I can share my screen. All right. Does that look good, everybody?
Looks great. Thank you. Go ahead.
Great. Okay. Well, thanks so much for having me at this conference. You know, we started our lab fairly recently, about 3 years ago, here in Seattle at the Institute for Systems Biology. And you know, I think that this is the future of the field in a large way. How do you kind of smash together these very large data sets on like host phenotype and what's going on in the ecology of the gut or the skin or other parts of the microbiome on the body? And there's 1,000 ways to do it, and there's no right or wrong way per se. It really depends on the question you're asking. So I'm not going to give sort of an objective view on what I think is the right way to do things. I'm just going to kind of go through a few concrete examples, because that's probably the most easily understandable way to sort of absorb some of this. So I'm at a Systems Biology Institute, and systems biology is inherently about integrating lots of different kinds of data from lots of different places, right? It's sort of the basic idea is the whole is greater than the sum of the parts, right? And it's only through having this integrated view or holistic view on the system that you're able to understand these emergent properties of the system, like health and disease, that you couldn't really get to from these kind of 20th century, drilling down on one system or one protein or one pathway type of approaches. So, you know, this intrinsically, I think the microbiome fits well into this paradigm. To give you an example of the kind of data collection paradigm I walked into when I came to ISB, this is just a little bit of a map of one of the clinical trials that's ongoing here on COVID-19. And you can see that, you know, there's a premium put on collecting a lot of information about a smaller number of people, right? The classical clinical trial to test a vaccine, for example, is to collect a small amount of data, like a few different things, for a very large population, say tens of thousands of folks, and you have really good statistics on that one factor. But oftentimes when you do something like that, one great example are small molecules for treating Alzheimer's disease. I don't know how many, hundreds or more have been tested, and everything has so far pretty much failed. And the problem with these classical trials is if you don't collect a lot of information about your system, at the end of the trial, if it doesn't work, you're sort of back to square one. You don't know where to go from there. And so, at ISB, one of the paradigms here is that you collect as much as you can about the system, so that if something fails, at least you don't have to start from nothing, right? You can sort of cut the population up into pieces and say, here's a group of responders, here are non-responders, and we can segregate them based on, maybe their blood metabolome or what have you. And that gives you an inroad to figuring out what's going on in the system. So when I came in, the challenge was, how do we slot the microbiome into this picture of dense host phenotyping? I don't think anybody needs much of an introduction at this point in the conference on what the microbiome is, but I do think that philosophically it's sort of shifted a bit how we think about ourselves, right, as meta-organisms that are more than just human, even though most of our biomass is, to be fair, produced through our own genomes. But about half a pound or so of our body weight is this mixture of hundreds of different species with a vast genetic repertoire, right? Millions, if not millions, even tens of millions potentially of genes pent up in this microbiome. And some of that genetic potential, we've outsourced some of the functionality of our bodies to. And understanding that is sort of the next phase in translating some of what we've learned about the microbiome into medicine. But I think, one of the fundamental challenges of translating microbiome science into medicine is making a link between the small molecules they're producing and what's going on in the body, right? The past 15 years of microbiome work has been really good at the sort of natural history of describing, who's where, under what conditions, what bugs are enriched in what disease state, or what populations of people have certain core taxa, what dietary and lifestyle factors are associated with certain organisms. That we've mapped out pretty well. There's a lot more work to do for sure along those lines, but what we don't quite know so well is how do we map all of that ecology into functional space? And I think the trick is the main sort of crosstalk happening between host tissues and the microbiota is in small molecules, right? There's this conversation going on between our microbiota and our bodies, and it's spoken in this language of small molecules, and we don't have a Rosetta Stone yet to kind of decipher what's being said. So we sort of have to start from the ground up and build up our understanding. But I do believe that the sort of crux of how do we manipulate ecology to affect clinical outcomes or health is bound up largely in deciphering this metabolic crosstalk. So the approach that we've taken for the past few years has been somewhat manifold here. One is very data-driven, so this sort of central part where we take a lot of information about the ecology of what's going on and say the gut. We mostly focus on the gut in my group. So metagenomics or transcriptomics or proteomics or what have you of the microbes, and integrating that information with really dense, even longitudinal phenotypic data coming from host organisms. And in particular, we've leveraged a data set that we have available here at ISB called the AFRL cohort, where there's about, I think it's about 5,000 people in general, but we have microbiomes for about 3,600 of them. And in addition to that, we have whole genome sequencing on these folks and blood metabolomes and blood proteomes and Fitbit measurements and questionnaires on lifestyle and diet. A lot of different information about this large population of people. So we can kind of ask, what amount of the variation in the ecology of the gut is actually resonant with variability in the molecular phenotype of the host organism. So that's a big push of our group, is sort of smashing together these data and seeing what that tells us. Ultimately, though, that's still all correlation. We're not doing any kind of intervention here, although I would like to get to that at one point in the future, but not yet. So we're just kind of looking at populations, looking at correlations. So to get beyond correlation, another approach that we've taken is to kind of build these mechanistic models these systems biology type models of the gut ecosystem, metabolic modeling to sort of ask, if you perturb the system, this really complicated and interconnected system, what are the outputs of that perturbation? And so I'll talk a little bit about that at the end of my talk here. And then tools for validating those predictions. So we can simulate these metabolic networks, but those simulations, how do we show they're true or not, or how well do they hold up under scrutiny? And we've developed these sort of poop soup experiments or ex vivo cultures, along with others in the field as well, working in this similar kind of in vitro model to directly validate the flux predictions from our metabolic modeling in these in vitro settings, and then potentially even intervention trials going forward. And if we have time, which I may not get to, there's a little bit of information on that. So this is largely in 3 parts. The first is like I said, this is all kind of concrete examples, not telling you like a strategy of how to do multi-omic integration, but just kind of giving you some things that we've done. This first story is our first paper that we published, where we're trying to sort of see whether or not we can predict something about the ecology of the microbiome that's imprinted in the molecular phenotype of the host organism. All right, and this is a art piece that Alison Kudla, a great artist at our institute, put together, where you're seeing kind of the starry sky with constellations of microbes in the shapes of their metabolites and the reflections of those metabolites in the blood red ocean. And I think that sort of metaphorical connection will be made here as I go forward. So this project was done with a couple of my really close collaborators here at ISB, Lee Hood and Nathan Price, 2 other faculty here. And it was driven largely by a couple of really talented postdocs, Tomasz Wolmansky and Noah Rappaport. And so the question is, can we predict this feature of the microbiome, alpha diversity, either how many species are there, the richness or the evenness, or some combination of those two factors? Can we predict this metric using information that we can measure in the blood of the host? And so the quick thing to do first was to ask, what omic is most resonant with this alpha diversity metric. We found, just to cut right to the main point, the metabolomics explained about almost half of the variation in alpha diversity. So you could sort of predict alpha diversity with this metabolomics signature, whereas proteomics in clinical labs, generally speaking, were very bad at predicting alpha diversity. And any combination of all 3 really doesn't do much better than the metabolomics alone. So metabolomics was sort of explaining the same variation that the other omics could explain as well. So sort of redundant to include other things. So we focused in looking at blood metabolites, untargeted metabolomics, measured through metabolome. So we built these lasso models, a sparse linear regression, this L1 penalty type model where you sparsify the number of features in it. We put in about 1,000 metabolites. That's what could be annotated and measured consistently across our population. And the model kind of ended up with about 40 with non-zero coefficients. So it was a 40 metabolite model. Most of them were lipids, but you also saw sort of amino acid derivatives and xenobiotics and steroids and things like that. The 11 metabolites that were most consistently associated with alpha diversity across all the different cross-validations and validation cohorts was this set here. And it was a nice sort of sanity check for us in the sense that these yellow-colored dots are indicating microbe host co-metabolites. So these are metabolites in the blood that are only there because of the activity of our microbiota. So for example, hippurate and cinnamoglycine are conjugated forms of small molecules like benzoate and cinnamate that are produced by the microbiome through breakdown of phenolics and complex polysaccharides that are in our diet. They go into the blood, they go to the liver, and benzoate gets glycine added to it and becomes hippurate, and cinnamate, same story. P-creosyl sulfate is a breakdown product that I believe tyrosine, but the microbiome kind of ferments tyrosine into P-creosyl, goes to the liver, gets sulfated, and that's what we pick up in the blood. And the lower 2 here are secondary bile acids, which are only produced because of the activity of the microbiome. So that made sense, but there are a bunch of other kind of host metabolites that were also detected here. In particular, this one was the sort of single most correlated with alpha diversity, and it's a derivative of testosterone. So this 5-alpha androsten 3-beta 17-alpha, it's a breakdown product of testosterone. It might be a purely host metabolite. It could also be that microbes are chewing on our sex hormones and breaking them down, but we don't quite know the genes involved in that currently. So, thinking about implications of this for health, we had data on questionnaire data on sort of health states of these folks. One thing we expected to see was that alpha diversity was sort of anti-correlated or negatively correlated with diarrhea. So the more diarrhea you have, the lower the alpha diversity is in your gut. And that was true both for the measured Shannon diversity from stool and also this M Shannon, which is kind of the metabolic Shannon that we could predict from our model. And looking at other phenotypic features that were significantly associated with alpha diversity, we found that abdominal pain, constipation, diarrhea were all significantly associated. Abdominal pain and diarrhea were sort of in the same direction. They were both negatively correlated with alpha diversity. So lower alpha diversity, you saw higher pain and higher diarrhea. Interestingly, though, constipation came up as positively correlated with alpha diversity. And if you think back to the prior plot with p-creosol sulfate, that's p-creosol is a fermentation byproduct of protein, and these derivatives seem to be kind of enriched in people who have constipation. So this sort of makes sense. But what we didn't really know is that there was this positive association between constipation and alpha diversity. So in this paper, we sort of discuss a little bit, is it a monotonic relationship between health and alpha diversity, as most folks sort of talk about when they're talking about it, or is it more complicated than that, which is what we suspect. I don't know if there's a Goldilocks zone or not, but some people with really high levels of alpha diversity are also very constipated and have high levels of these protein fermentation byproducts in their blood, which can be toxicants to, for example, the kidneys. One thing that broke our model was obesity. So if we looked at, the phenotypic imprint of obesity in the blood, we found that generally kind of inflammation markers were higher in obesity, but really only when you tipped into extreme levels of obesity. So this obesity 2-3 category. And if you fit our lasso model to different populations of people that fall into these different weight classes, generally speaking, normal weight to obese 1, the models looked identical, right? The same features came up as predictive in all those models, except for the obesity 2-3 group. So in that group, we sort of lost significance of this sex hormone derivative. And in addition to that, this is another picture of this, right? So you see the correlation between diversity and this sex hormone derivative in men and women, but it kind of goes away in this obese 2-3 group. In addition to the loss of significance of this particular feature, we also saw certain features became significant in this obese 2-3 group, and PFOS was a good example here. So PFOS is, I believe, in Scotchgard. It's known to bioaccumulate over time in our bodies, in particular in adipose tissue. So it made sense that perhaps if you have more adipose tissue, you would see an association here. And again, this is kind of showing the sort of loss of one signal and the gaining of another signal. So there's sort of a state change in the body. Maybe it's a leakier gut. Who exactly knows? We haven't quite found an answer yet. But this prediction between alpha diversity in the blood seems to shift only in the face of obesity. And a lot of the other things we tested, it was very robust too. One of the reasons we got into this paper to begin with, to predict alpha diversity, is because this prior association with alpha diversity and health. And in my thinking, I was interested because I saw alpha diversity as a risk factor for recurrent C. diff infection. There's a few papers sort of showing this, that if you are really low alpha diversity, you're at a higher risk of having recurrent infection. And the basic sort of theory around that is that commensals are kind of saturating the metabolic niche in the gut system, but as you lose diversity over time, over this loss of diversity, over this gradient, stochastically, you kind of start to lose these taxa that compete with C. diff for a metabolic niche. And so there's probably some transition across which, the sort of metabolic resources for C. diff to invade become available at low enough levels of alpha diversity. So we built a little 11 metabolite model to see if we could predict the lowest quartile of alpha diversity from the blood as perhaps a blood diagnostic that would be clinically applicable to sort of maybe segregate people as being higher or lower risk of C. diff recurrence. And it kind of worked. 0.88 is pretty good, but it's probably not good for a clinical diagnostic. So, more work to be done there. So that was the first story. We're sort of seeing the imprint of one component of the ecology of the gut in the bloodstream. So all these molecules that are bathing our organs and circulating all around our bodies, they're very resonant with what's happening in the gut. And that sort of tells us that it's very likely that the ecology of the gut is very relevant to a number of diseases and health states. So the next thing we looked at was not just across this kind of healthy population in Aryvale, but we wanted to look at an association with not a disease state, but a more interesting phenotypic state. And in this case, it's aging. And I'm plugging our recent press. We had this paper that was published about a month and a half ago, written about a month ago in the New York Times. So if you want a more pop-sci version of what I'm going to walk through in the next few minutes, you can check out the paper here. And this artist, Niv Bavarsky, made this really cool, beautiful picture of an aging microbiome. So to give a little background on this project, again, this was a collaboration between myself and Nathan Price and other faculty at ISB. And in addition, a guy named Eric Orwall, who's in Oregon, and he's associated with this aging cohort called Mr. Oss, about 1,000 older men who have kind of dense phenotypic data in addition to microbiome data. And Tom Womansky was sort of the driver of this, again, with NOAA, and a couple people from my lab, Christian Diener and Sushmita Patwardhan. If you look at the literature around aging in the microbiome, there's sort of two divergent responses out there. One is in centenarians, where you see this consistent drop in core microbes and a rise in subdominant taxa. But in people living in assisted living facilities in these other cohorts, you don't see that at all. You see almost the opposite, maintenance of these core taxa as you age, or even the slight increase. So we were thinking that this sort of disagreement in the literature might be related to multiple signatures of aging going on. So we thought we could tackle this with the data we had available. So we have this Aryvale data set that I talked about before, and it spans the whole lifespan. So from 18 to about 90 years old. Mr. Oss is this other kind of older cohort where people tend to be between about 80 years old and 100 years old. And for both groups, we have kind of dense phenotypic information in addition to some clinical information on health and microbiome measurements. And so one of the challenges here is that the prior work showing core taxa declining with age, these core taxa often shift depending on where you look throughout the world. So if I ask what's core in India versus what's core in Australia, it's going to be a different answer, probably Prevotella and Bacteroides respectively. So we wanted to have a metric that was sort of the inverse of core. So we developed this thing called uniqueness. It's a sort of beta diversity metric, and it requires you to kind of calculate your microbiome uniqueness in the context of a population. And it's sort of you can think of it as the sort of flip inverse of dominance of core taxa. As core taxa become less dominant, your uniqueness should rise because the organisms that sort of make us all more similar are declining in abundance, and the organisms that make us more unique are rising in abundance. If we look across all of our different kind of tests and validation cohorts, we saw that across the decades of life, there's this rise in uniqueness. So people's microbiomes are getting more and more unique as they get older and older and older, and it seems to actually kind of accelerate towards the latest decades of life. And just to give you a little more intuition about what this metric is, it's essentially your nearest neighbor distance to everyone else in the population. So you can imagine a sort of dimensional reduction where you project ecological distances on a 2D plane, say Bray-Curtis distances, and distances between these points is sort of how dissimilar two microbiomes are. And for example, this red point is more unique because it has a further nearest neighbor distance than this point here that's more central to this cloud of points that has a shorter nearest neighbor distance. So that's just how we calculated it. So what was interesting is that while people were kind of drifting apart from each other and becoming more and more unique, ecologically speaking, they were sort of converging at the metabolite level. So in the blood metabolome, we saw several metabolites that were significantly correlated with this uniqueness score. In particular, all these red metabolites are protein fermentation byproducts. Phenylacetylglutamine was previously patented as a marker of healthy aging, and it seemed to be highly enriched in the bloodstreams of centenarians. Not only that, though, it's a lot more abundant in the blood of the children of centenarians. So it's not only sort of a marker of having achieved extreme old age, but it's a marker of perhaps being likely to achieve that extreme old age. So that was sort of known. What was more interesting were these indoles. So we saw several of these indoles pop up. And if you look in the literature, indoles have been shown to causally increase both health span and lifespan in mice. So if you feed mice indoles, they live longer. And in particular, like the sort of mechanism behind that seemingly is that they interact with the aryl hydrocarbon receptor. They seem to be able to turn down inflammation and maintain or improve kind of gut barrier integrity. And indeed, these are made by the microbes in our gut. They're broken down from tryptophan into indole that gets absorbed and then travels to the liver and, for example, has a sulfate added to it. So this is indoxyl sulfate, for example. So, you know, this is all correlation. There's no causality here that we can say anything about because it's a population-based study. But at the very least, some of the hits that we're getting have previously in non-human animal models given us this signature, or have shown a causal signature with healthy aging. You know, again, thinking about this uniqueness score as the inverse of core taxa, in this Mr. Oz cohort, this older men cohort, it's largely Americans. And so the taxon that's the most core at the genus level across this group is Bacteroides. That's the most abundant core group in the guts of these folks. And it's strongly anti-correlated with uniqueness, right? So if your uniqueness is high, your sort of core taxon Bacteroides is low. So this is sort of the taxonomic signature of this uniqueness, at least within the population that we were studying. What was interesting, though, was that when we looked at this older population, the data I showed before was in AFRL, so that's 18 to 90 years old. We get this nice correlation between uniqueness and age, but this correlation was blunted, it was dampened when we looked at the Mr. Oz cohort. So we thought that perhaps this was due to the fact that there are indeed different trajectories of aging, a healthy and a less healthy. So we had to define health somehow. So Tomasz came up with 4 independent metrics. One was walking speed. So if you're a fast walker, you tend to be less frail and healthier as you age. Self-reported health, you know, either excellent or less than excellent. Medication usage, so we had a high group and a low group that we sort of defined. Life space score is just a score of how often you get around, right? How often you get out of your bedroom or out of your apartment or out of your neighborhood. And then composite healthy was you had to be healthy by at least 3 other metrics to be composite healthy. And when we broke people up into healthy or less healthy, we saw across the board that healthy people showed a pretty strong and significant correlation between uniqueness and age, and less healthy people did not. Thinking back on this core taxon signal, you know, we saw that in this composite healthy group, if you split them up into age tertiles, so T3 is the oldest of this older population, and T1 is the youngest of this older population, we see what we expect, which is a sort of steady decline in the prevalence of this core genus. And that signal is completely absent from the rest of the cohort, who are defined as being, you know, less than healthy. The sort of kicker to this analysis is we had follow-up data for this population. So there were multiple clinical visits, and there were about 4 years between the visits. And we had sort of baseline gut microbiomes for a fairly large population, and we had this 4-year in the future kind of follow-up. What we found is if you looked at uniqueness or if you looked at this core taxon, the story was the same. So if you had high Bacteroides abundance, which is sort of, you know, low uniqueness at the baseline time point, you were more likely to die at follow-up. And if you were in the lowest tertile of bacterial abundance, you were more likely to survive. And it was a significant effect. We got a little more resolved through a Cox proportional hazard regression model where we could kind of control for some of the confounders. And I'll just direct your attention to this lower right-hand quadrant. If you just look at 85-plus-year-olds, so people that are already quite old and that are close to achieving extreme longevity, and if you control for a bunch of different factors, including age, clinical site, batch effects, but then also we controlled for other factors that could influence mortality, like your cardiovascular health, your BMI, and a few other things. Even if you control for all of that, you still see that there's you're significantly more likely to perish at follow-up if you had high bacteroides or low uniqueness at the baseline time point. And this, if it's above 1 here, it means that higher bacteroides means you're more likely to die. And if it's below 1 here, it means higher uniqueness, you're less likely to die. So, even though this is a population, even though a lot of the associations we saw before were all kind of correlative, this is, if nothing else, diagnostic of survival, right? That people who show one pattern versus another seem to have different survival probabilities, and this is sort of robust to controlling for a lot of other factors that could be contributing to mortality. Okay, so my final part of the talk here, I only have a couple minutes left, so I'll just kind of breeze through this. We're also working on these mechanistic models where we can simulate an individual's microbiome. So postdoc in my lab, Christian Diener, really talented guy, built this flux balance analysis model. It's a sort of metagenome scale metabolic model that we can initialize with an individual's gut microbiome with all the abundances of their taxa. We can also initialize a personalized diet, and we can simulate the metabolic outputs of an individual's microbiome for a given dietary input. It allows you to predict a lot of different stuff. I'm going to race through this because I'm running low on time. One thing it allows us to do is predict response to dietary interventions. And so this is a very busy plot. I'll just quickly say that the first 3 rows here are acetate, the middle 3 are butyrate, and the bottom 3 are propionate. And this is the change in the flux of these different short-chain fatty acids for a given dietary input. So all the columns are different perturbations, either dietary, where you're turning up a metabolite, or probiotic, where you're turning up the abundance of a given taxon. And I'll throw your attention over here to a bunch of different dietary interventions that increase butyrate production in an individual who's metabolically healthy, but in 2 individuals who have type 2 diabetes, nothing happens. So you feed 3 people the same thing. One person, you see a big jump in butyrate production, and the other 2, you don't, right? So this is like a really cool argument for why you might need personalized diets. But this is all simulation. How does this actually match reality? Well, we can do experiments. So we can kind of redo our simulation in vitro with these poop soup experiments, where we take someone's poop and homogenize it and add individual components of diet to the homogenate and measure metabolic outputs. We did that for 2 different people, a sort of pilot study. These 2 people, given the same amount of inulin, produce different short-chain fatty acid profiles, no difference in acetate. There is an increased level in donor B of both butyrate and propionate production. And if we simulate from those same metagenomes what MyCom would predict for an inulin addition, the simulation agrees quite well with the measurement. So donor B produces more propionate and is predicted to produce more propionate, and same for butyrate, and there's really no big difference in acetate. So we're working on generating more data to sort of show that this is predictive across a larger population, but we're excited about the possibility of this as a tool for predicting metabolic outputs for specific inputs in a personalized fashion. And with that, I think I'm out of time, and I thank you all, and looking forward to questions after the next talk.
Great, thank you so much, Sean. That was a fantastic talk, and yeah, we'll take questions after the next talk. So with that, I would like to now introduce Keith Crandall, who's Professor and Director of Computational Biology of the Computational Biology Institute at George Washington University. His lab focuses on the development, testing, and application of bioinformatic and methodological approaches for studying microbial pathogenesis and gene variant associations with disease and treatment states. So thank you, Keith, and we look forward to your talk. Please go ahead.
All right, well, thank you very much. I need to share my screen. One, share. And hopefully that looks good to you. All right, so, all right, again, I'm Keith Crandall. I'm at George Washington University in Washington, DC. I direct the Computational Biology Institute at GW. And it's a pleasure to be here to talk to you about some of our methods. My lab historically has been method development and applications, and we apply things in a variety of areas to, well, natural history studies as well as human health studies. Our most cited piece of software is something you might have heard if you've done phylogenetics, something called Model Test that optimizes models of evolution for using in phylogenetic software with over 20,000 citations. We actually have, Susan talked about in the talk earlier today, these networking approaches. We have a network approach called TCS. This is a TCS network of over 7,000 COVID viral genomes that we put together early on in the pandemic just to get an idea what was going on. These are our newer software, Pathoscope and Telescope, are all about microbiome. And those are the two that I'm going to be focused on today. And I will say that, you know, my approach to software development is I love to think about new types of software and associated analyses with those kinds of software products. But I'm not so keen on, on software maintenance. So I love my graduate students and postdocs to develop software with me, take it away, and I never have to see it again. And in fact, this is what's happened, for example, with Model Test. My PhD student, David Posada, took it with him to Spain and developed a couple of additional versions. And now he's learned from me and done the same thing. And his graduate student, Diego, has now taken it on. So the latest version was just published a couple of years ago in Molecular Biology and Evolution Model Test NG. It actually, since he's in Stamatakis's group, this is the guy who writes the RAXML software for maximum likelihood and phylogeny estimation. So the Model Test NG fits very nicely into the RAXML framework now because of this collaboration. So I like to think about developing new methods, get them functional, and move them on to other people who do a much better job than I do at improving and maintaining the software products. And we've enjoyed a variety of support. And again, we have applications in basic science, organismal diversity like freshwater crayfish, especially around vision, and also human health. So I'm going to talk a bit more about our Pathoscope software to start with. So like Sean was just articulating in his talk, you know, the whole concept here is integrating different multi-omics data. And my colleague, Ali Ranavard from GW, talked yesterday about metabolomics and integrating metabolomics in particular with microbiome and his framework for doing that. We're going to use some of those same tests, but techniques, but now we're going to look at some general approaches to characterizing microbiome diversity, but also some different components of that diversity and some of the added things you can get out of that same omics data. So, and then look at associations with clinical data, because that's typically what we're interested in. So our Pathoscope software is out there in GitHub. All the code is available. We developed the initial underlying statistical methodology in 2014, and then had a follow-up paper in like 2016 or 17 with a modularized software product to implement. the approach. And the basic concept of Pathoscope is it's a k-mer-based approach, much like the approach that Rita Caldwell talked about earlier, where we just take a bunch of these raw metagenomic shotgun sequence reads. We create libraries from NCBI, and we map those reads against the library with bowtie 2, like all good bioinformaticians. We don't reinvent the wheel when it's out there rolling along reasonably well. But then our secret sauce is our Bayesian mixture model, where we look at mapping probabilities and remapping probabilities. So the problem with a lot of mappers is ambiguously mapped reads, where ambiguously mapped reads are treated typically in one of two ways. They are either mapped with uniquely mapped reads, in which case you don't get a lot of good resolution and differentiation, or they're tossed, in which case you're throwing away most of your data. So our Bayesian mixture model allows us to reassign those ambiguously mapped reads to partner with uniquely mapped reads and give us better differentiation. As a result, with shotgun metagenomics, we can actually get down to strain level differentiation, which is really nice for a lot of applications that we're particularly interested in. It's also relatively fast. The other nice thing is our library, unlike some of these reference libraries where they're kind of built into the software, we've now got a really nice module for building your own reference library. So you can, in updating your reference library. So we typically are using things like GenBank RefSeq library, or if we're particularly interested in specific microbes, we can add partial sequence data for those microbes. If we're looking at we've even run amplicon data. If we're looking at amplicon data, we can map it against either the RefSeq library or against green genes or one of those 16S based libraries. And you can also put in this reference library host information. So you can put in a host genome. So Adam Felipe gave a great talk earlier on in the session in this workshop on his updated human genome. I immediately sent off a link to that GitHub site to my students and said, let's update our human genome. And I'll show you why in a minute, because one of the first things we do is map, when we're sequencing, collecting sequence data from human samples, is map those sequences against a human reference genome, partition those aside, and then start focusing on the microbe reads. So that having a robust and detailed human reference genome becomes really important. So I also inserted this slide when I was listening to Susan give her talk this morning, or earlier today, because this is just for you, Susan. I wanted to give you the statistical model underlying this thing. This is a Bayesian approach that we do, where we're looking at proportions of genes mapping to different genomes. And then this is our secret sauce that gives us much better resolution than everybody else. Is this reassignment parameter that allows us to reassign based on information about how these reads are associated with uniquely mapped reads. We do take into account quality scores. So we love FASTQ files as opposed to FASTA files, and we can incorporate that quality information to our mapping score as well. So that's kind of what's under the hood, and gives us our strain level resolution with the metagenomics. So this is a typical kind of study that we work with, where this is a collaboration with folks at Children's National Hospital in Washington, D.C., where they have a bunch of kids who are asthmatic and controls. Used to be I had to explain what this swabbing thing was, but these days everybody knows what the swab looks like and what's going on here. So we swab these kids' nose and we sequence their snot, right? We're literally sequencing the kid's snot. In this case, we're doing RNA-seq. because we're interested in expression as well. So we can do metagenomics, we can do metatranscriptomics. And here we're interested specifically in identifying pathogens and immune response to that pathogen or those pathogens. So what our Pathoscope software does is take all these reads and then maps the reads to specific locations on reference genomes. So not only do you get information about what genomes in terms of your reference sequences are being expressed, but you also get stacks of reads on genes, on particular gene islands that are being expressed. So if you have high resolution, well annotated genomes, you can really get interesting information. So this is a typical, and in fact, in Amy's talk, she was talking about how there's difficulty of of all these things that can go wrong in a study, starting with the fact that in a nasal swab, you get about 90-10 partitioning of sequencing reads host to pathogens. So one of the big things we always worry about, especially when we start sitting down with collaborators who want to do one of these studies, is what material are you looking at, right? If it's fecal material, we typically see about a 50-50 ratio of host to pathogen reads. In nasal swabs, we see 90-10. We've sequenced from breast milk, and breast milk is like, 98-99% host with just very few microbes. So what this means is that you have to think about sequencing depth in a great degree, because you want to have enough sequencing reads to be able to actually characterize reasonably well the microbiome. So in this case, we have 90% host reads, 10% pathogen reads. The first step in Pathoscope is to differentiate those. We're going to focus on these 10% pathogen reads or microbe reads, and we want to go in there and identify whether there are pathogens or not. So we take those 10% reads, run them through Pathoscope. We get Pathoscope does abundance characterizations, and from that we can make calls on specific species down to strain level even, but specific species we identified here are the asthmatics and here are the controls. And as Sean was talking about, when you do these, it is everybody in the first day talked about who, when they're espousing the virtues of the 16S sequencing, this stuff is expensive, right? So you see many fewer individuals, but much richer data sets. So that's the trade-off. Here are our asthmatics and here are our controls, and we noticed this Moraxella cataralis in relatively high abundance in the asthmatics relative to the controls. We looked it up like all good bioinformaticians. Google is our friend. We looked it up on Google. It looked like a upper respiratory bacteria. So we reported back to our colleagues at Children's that, cataralis, Moraxella cataralis looked like it might be impacting. And they said, well, how do you know it's infective? And we said, well, we'll get back to you. So we had a lab meeting. We talked about it. We realized that, this is RNA-seq data. 90% of the data are the host. So let's just do a gene expression study. And we were fortunate in that another group had already published on host immune response to Moraxella cataralis expression and identified like 70 some candidate genes that were involved in immune response to infection by this bacteria. And so we looked at, because we had RNA-seq data, we could look at the expression of those genes. So these are all these candidate genes, and you can see a bunch of them are overexpressed in the asthmatics versus the controls. And so we called back up our colleagues at Children's and said, we have we can show that the kids are actually mounting an immune response, those that had infection with Moraxella. So another downside of the metagenomics data that was articulated on the first day is that it's a lot of data and it takes up a lot of memory. And the downstream analyses, a thing that a lot of people fail to talk about is the fact that the downstream analyses also take up a lot of data because they generate output files. So we regularly are struggling with our IT people to help them realize that, we actually need space to do these things. But the beauty of this kind of analysis is that you can really start getting at host functions, and you can start getting at real microbial functions because you do have RNA-seq data. So you can start relating what's going on with the microbes with what's going on with the host, and really have much more in-depth kind of insights into what's going on. So that's our Pathoscope software doing general microbiome characterizations with shotgun metagenomic data. It also works with 16S data. And the next piece of software I want to introduce you to is our Telescope software. It's specifically characterizing transposable elements and express transposable elements from RNA-seq data. And again, you have the same problem that most pieces of software out there that try and do this either map unique reads and throw away ambiguous reads, or they map them all together, in which case they get no differentiation. But using our same Bayesian statistical model, but now looking for those hallmarks of transposable elements, so 5', 3' LTRs with some open reading frame in between, we can identify transposable elements. And because we're mapping these RNA-seq reads, we can actually map to specific locations of transposable elements within the human genome because it's nicely annotated. So that's why I was anxious to get to that new genome, that updated genome data, specifically for looking at transposable elements. Transposable elements are of a growing interest in human health studies. This is just 2019. Kathy Burns and company talking about transposable elements in a variety of human genetic diseases. Of course, they were discovered by Barbara McClintock in 1980. Well, she won the Nobel Prize in 1983 for her discovery using maize, Indian corn, and the transposable elements are effectively what make these different coloring patterns. It was, of course, initially characterized as junk DNA, but actually makes up at least 45% of your human genome. I'll be curious to see with this higher resolution human genome if that number changes much, because of course it's a lot of these repeat elements that were not considered in the assembly of the current assembly of human genomes. And just a side note, Barbara McClintock is still the only woman to win an unshared Nobel Prize in medicine. So back in 1983, that's too bad. All right, so we presume that the transposable element expression is actually interrupting normal gene function and therefore has some, we think, cause implications for disease. As Sean was pointing out, a lot of what we do tends to be correlational, but we're developing approaches to try and get at that causation. So the first thing we want to do is the correlational study. So we have candidates for inference to put into the downstream more mechanistic approaches for studying. So we've done this with a few different diseases. We've done it with mental health diseases, with lupus here, and with various cancers. And I'm going to show you some lupus data and some cancer data. Lupus is an autoimmune disease that seems to transposable elements seem to be particularly impacting autoimmune diseases, especially certain classes. As we start analyzing more and more data, we're getting more insights on this. Lupus is mainly impacting women of childbearing years, and it's particularly predominant in minority racial and ethnic groups. There's no known cause. There are some implied genetic loci for this disease and some suggestions of HERV impacts. So we wanted to test this more thoroughly by looking at the HERV expression. So using telescope, we could characterize our HERVs, and because it's RNA-seq data and we get stacks of reads, we can look at expression, and we can look at, and we had, in this case, cases and controls, so we could look at differential expression of these HERVs, and we identify a whole lot of HERVs that are overexpressed and a few that are underexpressed that are going on. Now, we had 2 whole blood cell data sets that we looked at. Over 13,000 expressed HERVs, 321 of which are differentially expressed between the cases and controls. And we can, because again, it's RNA-seq data, we can also look at what's going on with the host genomics and look at differential gene expression of the host genes. So we can map those in terms of chromosomal locations and look at the host genes and look at the physical association of the HERVs that we're identifying with the host genes. So we can start making inferences, or at least predictions, about what genes these HERVs might be impacting in terms of their expression. And we can go further using some of Ali Ranavard's tools for pathway enrichment to really start looking at using some machine learning approaches to look at what pathways are being impacted by these associations between the HERVs and the host genes. And we've identified a bunch of these pathways in lupus. One is a response to type 1 interferon, one is a response to viruses. So this allows you, these techniques allow you to get more granular in terms of the pathways that are going on, that are being impacted. We've also looked at microbiome and cancer. And there are a lot of implications for microbiome and cancer. We worked on a review paper a few years ago. We looked at TCGA data, specifically at prostate cancer, breast cancer, and colon cancer. And these are wonderful data sets because they're RNA-seq data from a number of individuals from tumor tissue and normal tissue, adjacent normals. So you've got case and control right in the same data set. These average 70 million reads per sample, so they're really nicely, densely sampled. And this is just loading slow, so I pop past it. So we can do the same thing. We can look at HERV expression and how it predicts normal versus tumor tissue, and you can see there's clear differentiation. This is the prostate cancer and this is breast cancer. You can identify HERVs that are differentially expressed, overexpressed and underexpressed. You can look at those HERVs, where they are physically on the chromosomes and what genes are associated with those, and make those predictions. And then you can start, we're now starting to look at general HERV classes and their relative frequency and across different disease types and different, in this case, different cancer types. But we're also starting to collaborate. I've got a collaborator at GW, Kate Chapinelli, who actually has a nice cell model in cancer where she can go in and actually do the lab manipulations to express or not specific HERVs in relationship to specific genes, so we can start testing mechanistically what's going on with some of these. So it's a really robust system that allows us to go in and get detailed information, actually show mechanistically what's going on as we develop these hypotheses with our telescope software. So just a bunch of thanks. Evan Johnson's at Boston University. He's developing the Pathoscope software with us. Matt Bendall and Doug Nixon are Matt's developing the telescope. They're at Weill Cornell. Ali has the Omics Eye that he presented on earlier. Kate's doing the HERV knockout stuff in the lab to validate stuff. We've got lots of great graduate students, and even that cancer HERV analysis was done by an undergraduate at GW who's fantastic, and now in a bioinformatics PhD program at University of Chicago. So thanks a bunch.
Thank you so much, Keith. That was a wonderful talk. Really appreciate it. Thank you to both of our speakers. We will now open it up to the audience for questions. It looks like we have 2 questions in the Q&A. I'll go ahead and start with the first one. This is for Sean. The question is, did the Bacteroides abundance and survival coefficient also depend on the diet and exercise or other health factors?
It's a great question. We're honestly still trying to understand this decline in core taxon signal and how it specifically relates to health. We had some metadata on these older folks. We didn't have that much dietary information, so there wasn't a lot of useful information there. We definitely had information on their sort of physical fitness, but we didn't necessarily have exercise for these folks. But we had walking speed, we had how much they kind of got around in their lives. And for sure, this was associated with survival, but we try to do some controls where we had our various independent health metrics. And if you look within one, so for example, if I looked at people who take a lot of medications, so they're maybe less than healthy, but also within them kind of segregate people where some are, fast walkers and some are slow walkers. The fast walking high medication folks still showed an association with uniqueness and age. So when we zoom into these different metrics, it seems like the signal sort of split across all these different definitions of health. But it's a good question, and hopefully we can get more kind of detailed phenotypic data on future cohorts and understand better what these signals are telling us.
Thanks, Sean. The next question is for Keith. Based on your study of HERVs in lupus, have you started discussing with clinicians about using that as a diagnostic marker?
Yeah, we have. In fact, we work with a lupus company based out of Charlottesville, Virginia, called Ample Biosolutions. So they're very interested in developing some of these HERVs and associated genes as potential biomarkers. We also have a clinical collaborator at GW Hospital that is very interested in this. We really see these approaches as biomarker discovery tools, right? And so there's a lot of downstream work that needs to be done to validate them as reasonable biomarkers. But we think we definitely see both the HERVs and the HERV interaction with particular genes as combination biomarkers that could be very important for a variety of diseases. Like everybody, I reckon, we've been analyzing a truckload of COVID data in identifying similar sorts of HERV activity in the COVID patients as well that seem to help. Well, they're significantly associated with different disease outcomes, so we think that they could be informative biomarkers there as well.
Great, thank you. There's another question for you, Keith. Is there a significant difference in the annotation of metagenomes using your tools versus other tools such as Kraken? And can the observed disease correlations be reproduced with some of these other tools?
Yeah, great question. We've I took my slide out that shows our simulation study results. We basically took data from the oral microbiome project and used some Dan Hudson's simulation software to simulate sequencing reads of different links from those data, and then ran the data sets through in a bunch of different varieties, different sequence lengths, et cetera, through our software as well as a bunch of other software. Metaflan, Taxator, Kraken, and our software did very well. It outperformed everybody. Not statistically, Pathoscope and Kraken are very close. They're fairly different in how they actually operate, although they're both K-mer approaches, and they both performed very well. So I think things like Metaflan and and Taxator had a lot of false positives. So they could pick up everything, but they picked up everything, right? Including things that weren't there. So the Pathoscope and Kraken definitely had the best true positive and false positive rates and that paper is published in PeerJ, just came out at the end of last year. So that's available to take a look at. But they perform, yeah, our pathoscope performs pretty similarly to Kraken.
Great, thanks. Looks like we have a hand raised. Mike, do you want to go?
Yeah, thanks, Vanessa. So this is a question for Dr. Gibbons, actually. I'm sorry if I missed this, but where your uniqueness sort of comes in, are the unique taxa, are they similar between the older folks, or are they really just very different, those unique taxa? And there's a follow-up, I suppose. Do you think that those unique taxa are doing, thing throughout life, and you're just losing that core microbiome, but they're the sort of ones that are persisting? Sorry, 2 questions in one.
Great questions. So, okay, I'll try to quickly answer the first here. If you sort of paint out a PCOA of all these people in this population, it sort of looks like a C with like kind of two ends on it, with like dense groups of people on either tips of the C. And the 2 tips are defined by either having high Prevotella or high Bacteroides microbiomes. So, and the uniqueness score is sort of enriched at the kind of elbow of this C, which is associated with higher levels of the phylum Firmicutes in general. And I guess like maybe backing up a step, if you took 2 individuals' microbiomes and you compared overlap at the species level, say, you get like 30 or 40% of the species overlapping between 2 people. If you compare a third person, that drops to like, less than 10% overlap, and then it continues to drop further and further as you add more and more people. So very few taxa are like super prevalent across all people, but these are the sort of core, right? Like a lot of the Bacteroides species and so on. So yeah, are the subdominant ones the same across people? No, and that's why you get this rise in uniqueness. Everyone's got this kind of unique fingerprint or combination of these subdominant taxa that pushes them apart from each other as the core taxa decline in prevalence. We have some metagenomic data we've preliminarily analyzed, and it seems that like functional genes also sort of converge as the taxonomy converges, similar to what we see in the metabolome. And we do see genes involved in for example, indole synthesis or tryptophan degradation implicated. So there is even though the ecology is diverging, there's some kind of functional convergence going on. We don't quite know what's going on. We think that, this indole signal is interesting because it's been previously associated in an animal model as being associated with increasing lifespan. So maybe there's a causality link there where the microbiome is not just like hitching a ride and responding to aging, but it's also perhaps contributing to the phenotype, maybe. And another thing with these core taxa that we thought about is that a lot of these Bacteroides species can kind of switch their metabolisms between munching on mucus and munching on dietary polysaccharides. When we're young, we have a lot of goblet cells and we're just making mucus like crazy. And so maybe it's fine to have a lot of these taxa, even if we're having, say, a low fiber diet. But as we get older and our appetite declines and there's less throughput of both dietary polysaccharides and we're producing less mucus, maybe these same bugs that are perfectly healthy in a 20-year-old suddenly become a bit of an issue for an older person with a thinning mucus layer. So these are sort of the things we're thinking about what's the link between the phenotype and the signal.
Thank you very much.
Thank you. Sean, I had another question for you. Have you looked at the absolute abundances of microbes in the older population versus younger, and then specifically thinking about that for the Bacteroides? And then along with that, do you think that some of these changes is due to environment, diet, or both, or is it just completely unknown? Or I didn't know if you had any hypotheses on that.
Great question. And we don't have data like qPCR data, for example, on like total copies of 16S gene or something like that. So we're working off of either 16S or metagenomics for everything that I presented. So it's all, I guess, relative abundances in that sense. We're often doing like a CLR transform when analyzing the 16S, which sort of gets you a little further from the compositional issues. But we don't have absolute abundances. We've looked at absolute abundances in other studies, and we see cool stuff. Like one thing that could be happening with this decline in core taxa in older people is maybe the total biomass is like declining, and everything else is staying fixed, but some of these core taxa are reducing in their abundance. That could be happening, and we wouldn't pick that up because we don't have that absolute count data. We've seen in mouse models that if you sequence a microbiome of a mouse that's been hit by antibiotics, you see, oh, there's a huge enrichment in chloroplasts and mitochondria reads. Well, it's not that chloroplasts and mitochondria are blooming in these mice. If you do qPCR, you see that there's no biomass. Like if you get rid of all the bacteria, you're just sequencing the contaminants, which are these organelles, and they look in relative abundance terms like they bloomed, but it's just like a crash of the absolute biomass. So I would love to get that data from this cohort, but sadly I wasn't generating the data. I was kind of getting data from collaborators, but maybe in the future we'll try to get that.
Yeah, that's great. It's so interesting. So there's a question in the Q&A from Roland. Could the core taxa complexity reduction be driven by a simpler diet, say softer foods?
That's a good question. I'd have some insight into this because a friend of mine, when I did my postdoc in Eric Alm's lab, and a friend of mine, Thomas Gurry, who was another postdoc at the time in Eric's lab, did a feeding study where he switched a bunch of people onto an Ensure diet. So all these, I was in the study, and man, it was terrible. I had to drink Ensure for a week. I got depressed. Not eating solid food really sucks. But when you looked at the microbiomes of these people who all, I think 100 or so people switched suddenly from a solid diet to this Ensure diet, you would expect that everyone, the sort of the premise of doing this for this experiment was like spike-ins of individual dietary components and having a uniform background that everyone's eating. And you would think that everyone on an Ensure diet would make them converge somewhat in their microbiome profiles. Exact opposite happened. People actually drifted apart even further after they switched to Ensure. And we didn't quite understand why, but one premise is that Ensure is enriched for these simple sugars that perhaps flatten the fitness landscape somewhat, and you get these facultative organisms that are somewhat kind of putatively pathogenic that can grow on some of these simple carbohydrates that make us more unique, right? We kind of stochastically have these species across the population. So maybe the rise in those organisms made us drift more. So yeah, it's a good question, but it's complicated, and things, when we think one thing's gonna happen and we measure it, something else happens. So I don't think we understand it very well yet.
Yeah, for sure. So Keith, actually, I had a question for you with your Pathoscope tool. How well does that resolve different strains of the same species? And what if, do you have to have seen that organism before for you to be able to detect that? Can it do de novo detection?
Yeah, great question. In fact, in our initial publication on Pathoscope, where we described the Bayesian statistical model, we actually used as our data set, it was right at the time where there was an E. coli outbreak in Europe, and the Spaniards were getting blamed for it because of their cucumbers, and people were dying, and it was and there was a race to figure out what this thing was. And we analyzed the data. But when we analyzed the data with and without the group sequenced this new, it was basically a novel strain of E. coli, and they sequenced the genome of it. So when we first did the analysis without that genome, we had 85% of the reads mapped to what turns out to be phylogenetically the sister strain to that strain. So within all the breadth of E. coli, the thing that is sister to this new thing, 85% of the reads map to that, and the rest map to other E. coli. But when we included the sequence data from that new reference strain, and it wasn't fully assembled, it was a partial assembly, but we could include the partial assembly into our reference database, then 99% of the reads map to it. right, to that novel strain. So it's very sensitive. In fact, the whole methodology we actually developed, we collaborated with with Rita Caldwell and her Cosmos ID group back in the day, you know, after Tom Daschle got a powdery substance letter, and everybody was worried about identifying pathogens and then identifying which labs they came from. We collaborated on a project to sequence some of these genomes of some of the, you know, the Department of Defense top microbes of interest, of bioterrorism interest. So at the time I was at Brigham Young University, and it turns out we have a ridiculous collection of scary microbes. And so we wanted to sequence a number of them to characterize diversity and mutation rates, so we could tell whether we could actually, you know, how many generations will it take before you have enough variation to really tell whether you can distinguish between one lab being a source versus another and that sort of thing. But in the process, we got very interested in this question and started developing our own approach to look at these sequencing reads and differentiate quickly to identify pathogens. And that got us into trouble early on when we started using the software for, and that's the reason for the name Pathoscope, but that got us in trouble when we first started using it for general microbiome characterization, because it was a little too refined and started making some errors in terms of too much differentiation. But we tweaked some of the priors in the Bayesian model and got it kind of off that off that issue. So it's quite robust now. But yeah.
Awesome. So I guess I have a question for you both. So you did touch on, you know, thinking about the microbiome and the host and influences between them. And I wonder if you both could touch a little bit about how, you know, doing these multi-omic systems biology approaches, you know, how broad versus personalized they're going to be. And if you're thinking about taking into account the host genetics, so differences in different people's immune responses and things like that when you're putting together some of these models, and how this can really turn into real, maybe machine learning driven predictive approaches.
That's
Sean, if
certainly
you
where
want to
we're
take that
heading, and
first, Keith.
I'll take a stab at it first, and then let Sean finish up, and then we need to move on to the next session. But certainly that's where we're heading, is developing these deep learning approaches to integrate all this and to make predictions. So I think it's becoming very, very personalized medicine kind of approach that you may find some general trends. We're certainly seeing that with the HERVs, that there are certain classes of HERVs that are showing up repeatedly in autoimmune diseases in general. So I think there is some room for some generalizations. But, you know, at least where I'm very interested in going is developing approaches that really allow you to personalize things by integrating all this information. Sean.
Great answer, Keith. And it's a big question, right? I mean, I think there's 1,000 ways you could do all of this. And oftentimes a solution is quite bespoke to the question you're tackling, right? Is this involving the immune system? Is this involving metabolism? Like, what systems of the body are you trying to influence or predict? Generally, I think, yeah, you're totally right. There's I think two kind of main families of approaches. One sort of a very ML based approach, like data driven machine learning. Those models can be more or less opaque to interpretation. So they might be very good at predicting, but they might not give you a ton of insight as to like how or why. On the other side, there are mechanistic models. So I try to present some of that with the metabolic modeling, right? These are very transparent because everything is essentially accounted for, and you can know what every step is happening in the model and why. And that also allows you to do kind of personalization and integration of lots of data. So I think, you know, I think the future is probably some mixture of data-driven and mechanistic modeling.
Great, thank you so much. So again, thank you to you both. Wonderful, fantastic talks and stimulating session. And so virtual round of applause to Sean and Keith.